Hello and welcome to Professor Gadgil's GPAT Academy. Today we will discuss about general definitions and mechanism of drug absorption from GI. Biopharmaceutics is defined as the study of factors influencing the rate and amount of drug that reaches the systemic circulation and the use of this information to optimize the therapeutic efficacy of their products. Pharmacokinetics is defined as the study of time course of drug ADME and their relationship with its therapeutic and toxic effects of the drug. Simply speaking, pharmacokinetics is the kinetics of ADME. Clinical pharmacotherapeutics is the use of pharmacokinetic principles in optimizing the drug dosage to suit the individual patient needs and achieving the maximum therapeutic utility. Pharmacodynamics is the study of what the drug does to the body. Pharmacodynamic relates response to concentration of the drug in the body. Phases of drug administration are pharmaceutical phase, which includes the designing of the dosage form. Next is the pharmacokinetic phase. Third is the pharmacodynamic phase. And fourth is the therapeutic phase. Absorption is the process of movement of drug from its site of administration to the systemic circulation. Bioavailability is defined as the rate and extent of drug absorption. Any alteration in the drug's bioavailability is reflected in its pharmacological effects. Drug disposition can be simply clubbed as distribution plus elimination. Distribution is the movement of drug between one compartment and the other. Elimination is defined as the process that tends to remove the drug from the body and terminate its action. Elimination occurs by two processes. First is the biotransformation or the metabolism, which usually inactivates the drug, and second is the excretion, which is responsible for the exit of drug or the metabolites from the body. Ready to crack GPAT 2027? Join the study, dominate. This video covers mechanism of drug absorption from GI in detail, but one video won't crack your exam. A complete preparation system built. No confusing packages, just simple and clean GPAT mentorship. Download the GPAT Pro app, your all-in-one pharmacy exam companion. Ultimate Mastery for 5100, full course and all subjects. Crash course for 3100, fast track revision on important topics. MCQ Series 1600, 15,000 plus practice MCQs with solution. 3 modules free, start today, no payment needed. What's inside the app? 15,000 plus MCQs with solution, conceptual video lectures, to the point notes, daily revision cards, pharma job updates for government and private jobs, exam notices, latest pharma news, mnemonics, tricks and exam strategies, mock tests and performance analytics. Download on Play Store. Subscribe YouTube for more GPAT, NIPER and DI content. Join Telegram channel for daily MCQs and revision cards. Follow Instagram for educational reel content. Next, we will discuss the mechanism of drug absorption from GI. Drug absorption is defined as the process of movement of unchanged drug from the site of administration to the systemic circulation. Drugs that have to enter the systemic circulation to exert their effect can be administered by three major routes. First is the enteral, second is the parenteral, and third is the topical. Now we will discuss the gastrointestinal absorption of drugs. Movement of drug across the membrane is called as the drug transport. Inorganic ions and small organic water-soluble molecules like the urea can pass through aqueous-filled pores of perforations 4 to 10 Armstrong unit in diameter. There are three broad categories of drug transport mechanisms. They are the transcellular or the intracellular, in which there is passage directly across the GI epithelium. It is the primary route. Second is the paracellular or the intracellular, in which the passage is through the tight junctions or the persorption gaps. And third is the vesicular or the corpuscular, which is the energy-dependent cellular engulfment or the endocytosis. Now we will discuss in detail the transcellular or the intracellular transport. It is defined as the passage of drugs across the GI epithelium. The various transcellular transport processes involved in the drug absorption are: first is the passive transport processes. These transport processes do not require energy other than that of molecule motion or the Brownian motion to pass through the lipid bilayer. Passive transport processes can be further classified into following types: passive diffusion, pore transport, ion pair transport, and facilitated diffusion. First, we will discuss the passive diffusion. It is also called non-ionic diffusion. It is the major process for absorption of more than 90% of drugs. The driving force for this process is the concentration or the electrochemical gradient. It is defined as the difference in the drug concentration on either side of the membrane. Passive diffusion is best expressed by Fick's first law of diffusion, that is dq upon dt is equal to dAkmw upon h into bracket Cgit minus C. It is a downhill transport. The rate of transfer of unionized species is 3 to 4 times the rate for the ionized drugs. Drugs having molecular weights between 100 to 400 daltons are effectively absorbed passively. Permeability refers to the ease with which a drug can penetrate or diffuse through a membrane. Passive diffusion follows first order kinetics, that is dq upon dt is equal to Pcgit. Next is the pore transport. It is also called as convective transport, bulk flow, or filtration. This mechanism is responsible for transport of molecules into the cell through the protein channels present in the cell membrane. The driving force is the osmotic pressure, hydrostatic pressure. Water flux that promotes pore transport is called as the solvent drag. Compounds of molecular weight less than 100 daltons use pore transport. Generally, water-soluble drugs use pore transport. Chain-like or linear compounds of molecular weight up to 400 daltons can be absorbed by filtration. Third is the ion pair transport. Compounds like the quaternary ammonium compounds and the sulfonic acid forms the reversible neutral complexes with the endogenous ions of the GIT like the mucin and are transported across the membrane. Fourth is the carrier-mediated transport. The mechanism is thought to involve a component of the membrane called as the carrier that binds reversibly or non-covalently with the solute molecules to be transported. Carrier protein has always uncharged outer surface. Transport is structure specific. Drugs with structure similar to nutrients are absorbed. Number of carriers are limited and system becomes saturated and approaches asymptote. System has mixed order kinetics, Michaels-Menten, saturation or nonlinear kinetics. An area in which the carrier system is most dense is called as the absorption window. There are two types of carrier-mediated transport. First is the facilitated diffusion and second is the active transport. Active transport is of two types. First is the primary active transport, that is the ion transporters like the OAT, OCT, and ATP binding cassette. And second is the secondary active transport, that is the symport and the antiport. First, we will discuss the facilitated diffusion. It is a downhill transport and the driving force is the concentration gradient. Examples of such transport system include entry of glucose into the RBC and the intestinal absorption of vitamins B1 and B2 as well as vitamin B12. Second, we will discuss the active transport. This transport mechanism requires energy in the form of the ATP. It is divided into primary active transport and secondary active transport. Primary active transport is again divided into the ion transporters and the ABC or the ATP binding cassette transporters. And secondary active transport is divided into symport or the co-transport and the antiport or the counter transport. Now we will discuss the primary active transport, which is a subtype of the active transport. It is the process transfers only one ion or the molecule and in only one direction, and hence it is called as the uniporter. Second is the ion transporters. They are responsible for transporting the ions in or out of the cells. There are two types, the organic anion transporter, which aids the absorption of drugs such as pravastatin and atorvastatin, and the organic cation transporter, which aids the absorption of drugs such as the diphenhydramine. Next is the ABC or the ATP binding cassette transporter, which are responsible for transporting small foreign molecules like the drug and the toxins, especially out of the cells, and thus called as the efflux pumps, which make them clinically important. Next is the secondary active transport. In these processes, there is no direct requirement of the ATP. That is, it takes advantage of previously existing concentration gradient. The energy required in the transporting an ion aids the transport of another ion or molecule, that is, the co-transport or the couple transport, either in the same direction or in the opposite direction. The two types are symport or the co-transport, which involves the movement of both molecules in the same direction, and the antiport or the counter transport, which involves the movement of molecules in the opposite direction. Next, we will discuss the paracellular or the intercellular transport. It is defined as the transport of drugs through the junctions between the GI epithelial cells. There are two types. First is the permeation through tight junction. This process basically occurs through openings which are little bigger than the aqueous pores. Compounds such as insulin and cardiac glycosides are taken up by this mechanism. Second type is the persorption. It is the permeation of drug through the temporary openings formed by shading of two neighboring epithelial cells into the lumen. Paracellular transport involves the transfer of drug across the epithelium and through the cellular junctions. Third type is the vesicular or the corpuscular transport, that is the endocytosis. Like active transport, these are also energy dependent processes, but involve transport of substances within the vesicles into the cell. There are two types: pinocytosis, that is, cell takes the fluid along with the dissolved molecule, and phagocytosis. Orally administered Sabin polio vaccine, large protein molecules, and the botulism toxin that causes food poisoning are thought to be absorbed by the pinocytosis. Sometimes an endocytic vesicle is transferred from one extracellular compartment to another. Such a phenomenon is called as the transcytosis. Pinocytosis is also referred to as cell drinking. Next, we will discuss various comparisons. Energy required for active transport and vesicular transport. The direction for passive diffusion and facilitated diffusion is downhill, while for active transport it is uphill and varies for vesicular. Carrier is required for the facilitated diffusion and the active transport, as well as vesicle is required for the vesicular transport, but no carrier is required for passive diffusion. Passive diffusion is first order, facilitated diffusion is mixed order, active transport is mixed order, and vesicular transport is saturable. Typical target for passive diffusion is molecular weight of 100 to 400 and the lipophilic molecules. For facilitated diffusion, it is specific nutrients like the vitamin B12. For active transport, it is the ions and the toxins. And for vesicular, it is the large proteins and the vaccines. Clinical pharmacotherapeutics relies on this precise matrix. By understanding these specific cellular constraints, we can bypass the absorption barriers, optimize the individual dosages, and achieve maximum therapeutic utility for the patient. So, that's the mechanism of drug absorption in GI: passive diffusion, active transport, facilitated diffusion, and the pinocytosis. This is a high-yield GPAT topic. One question is almost guaranteed: master each and every bioform topic like this on the GPAT Pro app. If this video helped you, hit like, share with GPAT Buddy, and subscribe to this channel for more conceptual videos. I am Professor Gadgil. Keep studying, keep growing. See you in the next one. Jai Hind.